Hello and welcome to this video about COVID-19. As 6 biomedical laboratory research students, we are very interested in this ongoing pandemic. In this video we'll explain some things about the development of possible vaccines and the treatment of COVID-19. You must have heard of this new coronavirus because we can't get around it. In December 2019, several people in Wuhan, China contracted pneumonia of an unknown origin. In January 2020, the virus that was found responsible was identified as the SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome. The SARS-CoV-2 virus is a zoonosis, which means it can be transmitted to humans from animals. In the months that followed, millions of people around the world became infected. On March 11, 2020, the World Health Organization officially recognized the SARS-CoV-2 outbreak as a pandemic. When someone is infected with SARS-CoV-2, they will get the disease COVID-19, which stands for Coronavirus Disease 2019. But how do our cells get infected? The SARS-CoV-2 virus contains spike proteins called coronavirus S glycoprotein on its surface. With these proteins, the virus binds to the antikinase converting enzyme 2 receptors on the surface of the cell. With the help of an enzyme called TMPRSS2, the virus will enter the cell via endocytosis. The virus will then replicate itself in the cytoplasm of the cell by using the replication mechanism of the cell. When all of the different components are made, they are packed in a nucleocapsid with an envelope made from the intercellular membrane of the cell by a process called budding. The new virions are now complete and will leave the cell via exocytosis. When someone is infected with the virus, the incubation can take up to 2 weeks before showing any symptoms. This makes the virus spread easily without anybody noticing. Well, there are many ways to prevent it from spreading, such as wearing a mask, self-quarantining and washing your hands thoroughly. We need a vaccine to create a herd immunity much faster. So what can we expect from this new vaccine? Before we dive in deeper to the new corona vaccine, we must explain how vaccines work. A vaccine typically contains an agent that resembles a disease-causing microorganism that provides active acquired immunity. There are different types of vaccines, all based on a different part of a virus. One form of this is live attenuated, in which a virus is weakened in a laboratory. The virus can then replicate in the body, which is necessary to initiate an immune response and produce memory cells, but not so quickly that it makes the person sick. Another form of a vaccine is an inactive virus. This virus is no longer able to reproduce in the body, but it does stimulate the immune system. The vaccine then only contains polysaccharides or toxics. These are targets for the immune system. There is countless of research going on for creating a vaccine for the SARS-CoV-2. As soon as possible, most of the vaccines tested right now are based on the S protein. Some experts expect that the vaccine will be ready within a record-breaking time, 1 to 2 years. Usually, the process from testing to distribution of a new vaccine can take up 6 to 10 years. This is often a very long process because of the many steps to ensure the safety and effectiveness of the vaccine. There are different phases to testing a vaccine. First, it is usually done in living cells in the laboratory, then in animal experiments, and finally the new vaccine is tested in humans. The last process is the most time-consuming and very thorough. With the new SARS-CoV-2 vaccine, they needed to start at the very beginning of the process. Though there were other deadly coronaviruses, a vaccine for those viruses was not considered necessary. The SARS-CoV-2 is more contagious and deadly than the previous ones, which urges to make a vaccine as soon as possible. There will be some side effects. The trial volunteers have been warned that some may get a sore arm, headaches, or fever in the first couple of days after vaccination. An example of a vaccine in development is from the clinical research team at the University of Oxford Jenner Institute and Oxford Vaccine Group in the UK, who seem to have an early lead. Researchers began working on a vaccine in February, and human trials started at the end of April. The vaccine is made from a weakened version of a common cold virus from chimpanzees, which then has been modified so it cannot replicate in humans. The spike glycoprotein from the SARS-CoV-2 have then been added. This is done in the hope of making the body recognize and develop an immune response to the spike protein that will stop the virus from entering the cells and therefore preventing infection. While vaccines are one way to stop the virus from spreading, we also need to focus on the treatment of patients who already are infected. There isn't yet a specific treatment for COVID-19. The treatment of seriously ill people consists of treating the symptoms. For example, doctors can give extra oxygen to patients who are very short of breath. Painkillers can help with complaints of fever and sore throat. A virus can be killed because technically it's not alive, but the multiplication of viruses can be slowed down by using virus inhibitors. An example is remdesivir. Remdesivir acts as a nucleotide in the body. SARS-CoV-2 uses an RNA-dependent RNA polymerase for transcription. This viral polymerase struggles to distinguish the virus inhibitor from true nucleotide. When the remdesivir is built in, further transcription is blocked. This hinders the virus to multiply. Remdesivir is currently still in the experimental phase, but the drug has shortened the recovery period in 31% of the patients. However, it appears that virus inhibitors can have serious side effects, like heart problems. We hope that we have clarified some things about this virus, the development of a vaccine and the treatment of COVID-19. We hope you enjoyed the video and thank you for watching.